All right, I am ready to start making my gel. I have all of the things that I need here, at least to start before the microwave goes. So I have this gel box here that has in it this caster or mold, and it has this sort of rubber, flexible rubber on the outside, and it will eventually run this way. But for now, we're going to put this in here perpendicular to the way it will run. And you want to make sure that the rubber here does not move because it started to. And you'll put this into the gel box, and it makes a really firm seal on either side. So we can pour the liquid gel in there once it is microwaved and ready. So I have already measured out a gram of Agarose, and this is just a white powder. I'm gonna put that into my flask. And I have 100 milliliters of this 1x TAE. So I've already diluted it from a 50x stock solution. All right, and we can see that it is this opaque liquid to start. I'm gonna now put this in the microwave, and I'll be back to show you what it looks like. We have now this clear sample, right? That's our gel that's completely mixed in there and melted. If you have any little bits that are floating around, probably want to pop it right back in the microwave. But we're ready, and it's cooled, and so I am now ready to add the ethidium bromide to this. This is our carcinogen. This is why I'm wearing gloves. That's why also we have these flasks that are labeled. Add the ethidium bromide to this. This is a pipette that's only used for ethidium bromide, and then this pipette tip will go in the hood. I'm going to swirl this up and pour this into that mold. And then add our comb in here. We're going to use a comb that has 6 wells in it. And then we will let this sit until it polymerizes. It is now really clear. When it polymerizes, it will look really opaque. I'll show you what that looks like when we get there. All right, we are ready with a solidified gel that we can now run those PCR products. They have been taken out of the thermocycler, so you can see that that gel is a little bit hazy or opaque, which suggests it is now polymerized. It is also hard to the touch. Again, this contains ethidium bromide, which is a carcinogen. So anytime you're working with Agarose gel electrophoresis, you'll want to make sure that you're wearing gloves. Okay, so I'm going to take out the comb. You can do this before or after you take the gel out. I use both hands on either side and pull it just straight on up. And then I'm going to wiggle this gel out of the caster and then turn this so that it's now in the buffer. And then pour some more buffer, 1x TAE, on top. And we do use the same buffer both in the gel and in the chamber that the gel is in. That's important in order to have essentially the same salts and pH that are running through the gel and conducting that electricity through the gel. So now we can load our sample. I have our tubes here from PCR in our ice bucket, and I also have some tubes that I'm going to pipette those samples into and mix up the loading dye with them. And now I'm going to add this purple loading dye. It is 6x in that it does need to be diluted to 1x, so we are going to do that. This loading dye is used because it does have dye that's going to be seen in the gel because DNA is clear, so we wouldn't be able to see it otherwise as it moves through the gel, so we have an idea of how far that DNA has progressed through the gel. In addition, it has glycerol in it, and that will help weigh down our DNA sample so that it can easily fall into the well that we pipetted into. So I'm going to add some loading dye. So you have that dye in all of our tubes. And now we just need to add the sample to it. And again, know what you're loading into each of those tubes so that you don't lose track. So it's always good to sort of label the tubes ahead of time, even have a cheat sheet written down on your paper so we know what is what. So the samples are now ready to load. I have them in those little PCR tubes again. Let me show you what they look like. Seconds. I'm actually going to vortex it quickly to mix it up. So we have our samples spun down and ready to be loaded. We do have to load a ladder, so I'm going to skip the first lane and start loading this in. So I'm going to put the pipette tip just over the well, and that sample will just fall right into the well. You can see that there's a little bit of purple in this well right there. Sample 2. Sample 3. So those are all loaded. Now I want to do the ladder. This ladder, it depends on what sample that you're using, that you might want to consider different types of ladders. So this DNA ladder is going to be used as a reference. It again just looks like the bluish purple color because it has that dye in it. And I will provide the ladder to you so that you can see this as a reference. So these contain DNA fragments of known lengths that can be used as comparisons. So I'm going to load that ladder in there. And everything is where it needs to be. So now consider the charge of DNA. DNA being negatively charged means that we would want to run that DNA towards the positive electrode over here. So you can see that I have the wells on the opposite side. So they're going to move in this direction. I have this hooked up to a power supply. So I'm going to turn this power supply to a higher voltage. About 120 for this device is fine. This does depend on the equipment that you're using. And I'm going to go ahead and press start. And what you may or may not be able to see is that bubbles start to form. So hopefully if you look closely, you can see these little bubbles forming. Those bubbles are a good sign that we have electricity flowing. So we wait about 30 to 40 minutes. It's good to check the place of that dye front as it moves through the gel. So you can see things at the top. You can see where those wells are. That's where our dye is. So we'll come back later to see that dye moving through the gel, moving towards that positive electrode, and then image the gel under a UV transilluminator. So this machine over here is a transilluminator. It has a UV light inside that can be used to visualize the gel. Our gel, which I'm holding in a gloved hand because that ethereum bromide, you can see that purple dye down there. It has gone about 3/4 of the length down the gel. So our sample should have also gone that far. And so I'm going to put this in the transilluminator and take a picture of it. So this is what the transilluminator looks like when the door is opened and the gel is inside. And I'm going to shut that and start the imaging on the computer that you see on the other side of the gel box. I captured an image of our gel here, and it looks like we have PCR products, which is wonderful, and I will download this file as a TIFF file so that you can look at it on your computer and incorporate it into your lab report. The last thing is that I want to dispose of our waste properly. So since this gel has ethereum bromide in this, there's special waste in the hood where that ethereum bromide waste will go.